I'm Hisako Akiyama from Riken Center. Riken Center for Brain Science in Japan. Today's my talk is entitled Receptor of Shiga.
Your microphone is muted. Your phone is turned off.
I'm sorry. So I start again. I'm very sorry about that. And I'm Hisako Akiyama from Riken Center for Brain.
It's muted again. We can't hear you.
Can you hear me?
Yes, now yes.
Can you hear me? It's okay.
Yes, now we can hear you.
Thank you very much. I'm sorry. I'm Hisako Akiyama from Riken Center for Brain Science in Japan. And today's my talk is entitled Receptor of Shigatoxin galabioceramide is present in human cerebrospinal fluid. Shigatoxin is a toxin produced by enterhemorrhagic E. coli 0157 and Shigella dysenteriae. An infection with shigatoxin producing E. coli causes bloody diarrhea and hemolytic uremic syndrome, so-called STEC-HUS, and shigatoxin binds to galactose alpha 1,4-galactose beta structure, and shigatoxin binds to cells expressing compounds with structure and inhibits their protein synthesis. And about 30% of patients with STEC-HUS develop central nervous system CNS complications resulting in neurodegeneration and demyelination. And CNS complications are the main cause of sudden death in STEC-HUS. And it has been reported that the binding of shigatoxin to a glycolipid, globotriaosylceramide, GB3, causes the CNS complications. And GB3 has this structure at the terminal of its oligosaccharide chain. And today's topic is ceramide dihexoside, CDH, in cerebrospinal fluid. We discovered this lipid by hydrophilic interaction liquid chromatography tandem mass spectrometry, HELIC-MS-MS.
Sorry, doctor, we cannot see your slides. We can see the first slide only. Are you moving in the slides?
Yes.
Okay, now. Yes, now we can see.
So can you see my slides now?
Yeah, okay. Now it's the second slide. Yeah.
Okay. So I would like to explain again. I'm sorry. And shigatoxin is produced by enterhemorrhagic E. coli, like L157 and Shigella dysenteriae. And shigatoxin binds to cells expressing galactose alpha 1,4-galactose beta structure. And infection of shigatoxin causes bloody diarrhea and hemolytic uremic syndrome, so-called STEC-HUS. And 30% of STEC-HUS patients develop central nervous system CNS complications, resulting in neurodegeneration and demyelination. And it has been reported that glycolipid, globotriaosylceramide GB3, causes the CNS complications. And GB3 has this structure at the terminal of its oligosaccharide chain. And today's topic is ceramide dihexoside CDH in cerebrospinal fluid. And we discovered this lipid by hydrophilic interaction, liquid chromatography, tandem mass spectrometry, HELIC MSMS. And I'd like to briefly introduce the HELIC mode. And in the HELIC mode, compounds are distributed by hydrophilic interaction, and water-rich layer is formed on the highly polar stationary phase like silica gel, and compounds are distributed between the water-rich layer and mobile phase. And HELIC mode enables to separate a glycolipid by difference of their carbohydrate structure. Therefore, the HELIC is widely used for separating isomeric glycolipids, for example, glucosylceramide and galactosylceramide. And lactosylceramide and galactosylceramide are members of ceramide dihexoside, CDH. And when we analyzed CDH in lipid extracts from human cerebrospinal fluid, CSF, we detected two peaks. And as this paper has already reported, lactosylceramide and GB2 can be separated by HELIC. As the presence of lactosylceramide in CSF has already been reported, however, the presence of GB2 in CSF has not been reported before, and we speculated that these two peaks might be lactosylceramide and GB2, and then we decided to purify CDH from CSF and analyze its structure. And in our study, for CDH purification, we chose CSF from patients with idiopathic normal pressure hydrocephalus. In this disease, CSF abnormally accumulates in the brain, and as a part of treatment, CSF is removed, so we could obtain a large amount of CSF at one time. And lipid extracts from CSF were subjected to normal phase chromatography using chloroform methanol gradient. And this is a result of TLC analysis of the CDH elution profile from a silica gel column. And around fraction number 19 to 25, there were very faint bands that showed similar RF value with CDH standard. And HELIC MSMS detected two peaks in the purified fraction number 19. And this graph shows the fatty acid composition of peak 1 and peak 2 from fraction numbers 19 to 25. Although the peak area of peak 1 was greater than peak 2, but their fatty acid compositions were similar. Next, we conducted immunoblot analysis of the purified CDH fractions using shigatoxin subunit B. And shigatoxin has subunit A, a toxic domain, and B, a receptor binding domain. And subunit B binds to a galactose alpha 1,4 galactose beta structure. And GB2 and GB3 both have this structure at the terminal of their oligosaccharide chain. And this is a result, and purified CDH fractions show the positive reaction with shigatoxin subunit B. And this indicates that in the purified fractions, there might be compounds with this structure. And this is this figure shows the synthesis and degradation pathway of GB2 and lactosylceramide and GB3. And glucosylceramide is synthesized from ceramide by UGCZ, and lactosylceramide is synthesized from glucosylceramide. and GB3 is synthesized from galactosylceramide by ethyl-gal-T. And galactosylceramide is synthesized from ceramide by CZT, and GB2 is synthesized from galactosylceramide by ethyl-gal-T. And because GB2 standards are not commercially available, to identify the two peaks detected in the human CSF, we used HeLa cells genetically engineered to express different glycosphingolipid metabolizing enzymes. Parental cells and ethyl-gal-T knockout and UGCD knockout and UGCD double knockout HeLa cells were established by Dr. Yamaji at the National Institute of Infectious Diseases in Japan, and this table shows the predicted expression patterns of glucosylceramide, galactosylceramide, lactosylceramide, GB2, and GB3. And here are the results of glucosylceramide and galactosylceramide, and the results were consistent with the predicted expression patterns. And this is the result of peak 1 and peak 2. And this chromatogram shows the CDH having C24-1 fatty acid in ceramide part. And this graph shows the combined value of CDH having other lengths of fatty acid. In the parental HeLa cells, two peaks were detected, and these two peaks have similar retention time with these two peaks detected in purified fraction from CSF. And the peak 2 was not detected in ethyl-gal-T knockout, and peak 1 was almost undetectable in UGCD knockout, and both peaks were almost undetectable in UGCD-CZT double knockout cells. In addition, peak 1 was increased when the lactosylceramide standard was added to the purified fraction. From this result, we conclude that peak 1 is lactosylceramide and peak 2 is GB2. This is a result of CDH having C25 and 25 fatty acid in ceramide part. In the parental HeLa cells, in addition to peak 1 and peak 2, another peak, peak 3, was detected. A peak with similar retention time of the peak 3 was also detected in purified fraction from CSF. In the helic mode, glycosphingolipid that has fatty acid, hydroxylated fatty acid like this in ceramide part retains stronger compared to that having non-hydroxylated fatty acid in ceramide part. And the molecular weight of C25CDH is the same as C24-1 hydroxylated fatty acid CDH. So we speculated that peak 3 might be C24-1 hydroxylated fatty acid CDH. And this is a quantification result of peak 1 and peak 2 and peak 3. And the expression pattern of peak 3 was similar to that of peak 2, but was clearly different from peak 1. So we speculated that peak 3 might be hydroxylated fatty acid GB2. This is the result of GB3. Having this chromatogram shows the GB3 having C24-1 fatty acid in ceramide part. And single peak was detected in the parental HeLa cells, but not in ethyl-gal-T knockout and UGCD knockout and UGCD-CZT double knockout HeLa cells. And also, GB3 was below the detection threshold in fraction numbers 19 to 25, isolated from CSF. And similar results were obtained in GB3 having another length of fatty acid. As I already showed today, immunoblood analysis show the positive reaction with the cigatoxin subunit B in the purified fractions from CSF. And these GB3 expression indicate that the GB2 but not GB3 mediated the cigatoxin subunit B positive reaction. Based on our result, I would like to discuss the origin of GB2 detected in CSF. About 80% of the CSF composition is from blood, so GB2 synthesized in the tissues other than the brain might be incorporated into CSF. The remaining 20% may be synthesized in the brain, and there are two possibilities for the production site. The one is myelin. In our study, we detected the GB2 having hydroxylated fatty acid in ceramide, and hydroxylated ceramide expresses in myelin, epidermis, and small intestine, and in the brain, galactosylceramide is synthesized from ceramide by CGT in oligodendrocyte. As FOGLT expresses both in neuron and myelin, GB2 might be synthesized in myelin. And the other candidate production site is the choroid plexus, the site of CSF production. As FOGLT and galactosylceramide both express in the choroid plexus, GB2 might be synthesized there and is released into CSF. And finally, I would like to discuss a relationship between GB2 and STEC-HUS. And GB3 has been reported in neuron but not in glial cell. And GB2 and GB3 both have galactose alpha 1 galactose beta structure at the terminal of the oligosaccharide chain. And as I introduced at the beginning, binding of cigatoxin to GB3 has been reported to cause the CNS complication in STEC-HUS. As GB3 expression is found in neuron but not in myelin, I think it is difficult to explain why demyelination occurs in CNS complications in STEC-HUS. In our study, we found the possibility of the presence of GB2 in myelin, so I would like to propose that the CNS complications in patients with STEC-HUS are mediated not only by GB3 but also by GB2. And it will be very important to determine whether GB2 is really expressed in the brain. And finally, I would like to thank all the members who have joined in this work. Thank you for your attention, and very sorry to have technical problems with my slides. Thank you for your attention.